Hello, good morning, good afternoon, good evening, from whatever time you'll be watching this video from. I want to welcome you to yet another exciting lectures on biochemistry simplified, and we've recently been looking at biotechnology and the genome with respect to genetic engineering. And I welcome you especially to the continuation of this lecture. So we'll continue just right from where we left off. And this moment we'll be looking at the isolation of DNA. The isolation of DNA. Okay, so we've looked at pretty much about the brief history of biotechnology, what is used for, how it's been used, the DNA in question, identifiers like the genetic markers, and now we want to look at the isolation of DNA. So let's just jump right in. DNA is usually found in the nucleus of cells. Okay, and cells are the fundamental units of life. So all organisms are actually made up of cells, which could either be prokaryotes or eukaryotes. Whatever the case, DNA is usually found located either freely in the cytoplasm, that's for prokaryotes, or in the nucleus for eukaryotes. So that will make us look briefly at the prokaryotes and eukaryotic cells. Okay, so let me note that down, that DNA in question, before you can isolate anything, it's important to know the location of what you are isolating it from. All right, so DNA is usually found in the nucleus of cells. The nucleus of cells. Okay, great. And cells in itself are the fundamental units of life. Okay, so next important point is that the cells are the fundamental unit of life. All right, and all organisms are made up of these cells, which could either be prokaryotes. All organisms are made up of cells which could either be prokaryotes or eukaryotes. So all cells can either be made prokaryotes or eukaryotes, all organisms. All right. So whatever the case that it could be, whether prokaryotes or eukaryotes, it's important to know where they are located, because from where they are located, you can now begin to talk of how to get to them and isolate them. So whatever the case, that is whether eukaryotes or prokaryotes, DNA is usually found located either freely in the cytoplasm, cytoplasm, in which case you could call them prokaryotes, or they are found in the nucleus, or in the nucleus, and in which case, let me do this here, in which case you can call them eukaryotes. All right. So prokaryotes and eukaryotes. So in eukaryotes, so if we start with, let's take prokaryotes first. So in prokaryotes are spotlight here. So in prokaryotes, what you found is that the nucleus or the nucleoid consists of the bacterial DNA and is enclosed within a membrane. Okay, so in prokaryotes or prokaryotic cells, the nucleoid a nucleoid consists of the bacterial DNA, consists of the bacterial DNA and is enclosed within a membrane and is enclosed within a membrane. All right, so that is for prokaryotic cells. Okay, so I want to quickly give a sketch of what the prokaryotic cells look like. Okay, let me use green here. So prokaryotic cells could assume this kind of shape. All right, so if I double that, sorry, okay, so in prokaryotic cells, then you have the nucleoid that is enclosed. Okay, something like this. Okay, so we say the nucleoid is enclosed within a membrane, and what you have here is what? Your nucleoid that is enclosed within a membrane, and the membrane could be something like a subsection. Sorry, let me do that in black or blue. Okay, so purple will be fine. Let me. Okay, so we have the membrane looking like something like this. So you have this segment here or section here. Okay, and this could represent the membrane. Okay, the whole of this like this. Okay, could represent the membrane, and um, that includes the inner plasma membrane, the cell wall, the perisplasmic space, and what's the outer membrane. Okay, so this could further be expanded this way for clarity. Sorry about that. So for clarity, you could have something like this. Okay. Okay, so the whole of that membrane here, then you could have like a dark area. Could do this shade. So all of this is kind of representing what the membrane is, a dark area, okay, and you have um, dark area. Let me see what color I do it here. Semi space. Okay, so I'm just using different colors to represent different segments of the membrane. So in that case, this particular one here is too far apart, so you have this forming right here. Okay, so I could now extend this as coming like this to represent the membrane. So this area or this line right here could be referred to as what? Your inner plasma, your inner or your plasma membrane. Alright, and you have this inner wall here, that represents your cell wall. Okay, then you have those inner space here. Okay, this inner space here that represents your periplasmic space. Periplasmic space. And of course, you have here representing your outer membrane. Okay, so that is a sketch of the prokaryotic cells. We try to sketch it as simple as possible, and the whole of the membrane that the nucleus is enclosed in contains different aspects such as the inner plasma membrane and the cell wall, periplasmic space, and the outer membrane. Okay, so that is for the prokaryotes. However, for the eukaryotes, which we'll be looking at shortly, okay, so for the eukaryotes right here, okay, so for the eukaryotes, what we'll have is that the DNA, okay, so however in the eukaryote, the DNA is found located within the nucleus. Is found located within the nucleus. Okay, so it's found located within the nucleus. All right, so I want to also look at like a form of a sketch as well, where we could locate the DNA of eukaryotic cell. Can I do this here? Okay, so it's found located within the nucleus. Let me take black. Okay, and right here you could have a double thin line. Alright, so within the nucleus, within the space of the eukaryotic cell is found nucleus. Then you have other components that flows okay from the nucleus. Then you have okay okay and you have bodies like the Golgi vessels. So this is like a schematic sketch of the eukaryotic cell. All of this is important because for DNA isolation, you want to reach out to isolating and getting out the DNA from the nucleus. Okay, so this is key. All right, so maybe I should use different color to indicate what here. Okay, I have a yellow color. Sorry about that. Okay, and you have a white space of sky blue all over the nucleus. So more like a schematic sketch. So understand that this is like an illustrative diagram kind of. So right here you have the nucleus. Okay, then this is your mitochondria. So this, just like we saw a schematic stretch and sketch mitochondria naturally, a schematic sketch of the, and here you have the lysosomes. Then these small circles around here is the sensory vesicle. Okay, then of course you have the rough endoplasmic reticulum right here. Rough endoplasmic reticulum. Alright, then you have right here the nuclear membrane. The nuclear membrane. Okay, then we have this as the Golgi bodies or the Golgi bodies. Golgi vesicles. Alright, then of course, I go to this here. We have the plasma membrane. Okay. What else? You have the perisosomes. Okay, so I should there's the perisosomes. Peroxisomes. Okay, so this is a schematic sketch. Let me put one more color effects here. So this is like the perisosomes. Okay, that's fine. So this is a schematic sketch right here of the eukaryotic cells, and we say that the DNA, which is of interest, okay, is located where? In the nucleus. So we have to now look at ways in which we isolate this DNA by breaking up the cells and getting through to the DNA. Okay, so other constituents of the nucleus includes, okay, so note, it's not just the nucleus contains other things. All right, so worthy of note is that other constituents of the nucleus other constituents of the nucleus include the following uh, includes the following. Okay, and one, you have the ribonucleic acid, which is your RNA. Okay, then you have the proteins still in the nucleus, the proteins, then you have the carbohydrates. All right, then of course we have the lipids, among others. Okay, etc. All right, then for this isolation to take place, the first necessary step is the disruption of the cell. That is very key to note. For the destruction to take place successfully. The first necessary step is the disruption of the cell to release either the nucleoid or the nuclear materials. So thus, having known all of this, for the isolation of DNA, the first necessary step is the disruption of the cell to release either the nucleoid, case of prokaryotes, the nucleoid or the nuclear material. Case of what? Eukaryotes. So that's very key. On our next page, we'll now begin to look properly at what the DNA isolation. So what we've been able to achieve right here is that we got the structure of the prokaryotic cell, the schematic diagram, and we also looked at the schematic structure of what the eukaryotic cell. And with their component parts, and we saw that for the isolation of DNA to take place in the first place, what you need to do is to break the cells, the disruption of the cells. So in our next page now, we'll be looking at what exactly is DNA isolation and how do we go about DNA isolation. Next slide. Okay, so what exactly is DNA isolation? We say that DNA isolation, therefore, DNA isolation, therefore, refers to the process of extracting DNA. Okay, that's the focus. We want to get the DNA. So the process of extracting the DNA from a cell in a relative pure form, in a relatively pure form, not as crude. We want to get it as clean as possible. Okay, so it therefore involves separating the DNA from other cellular components. It therefore involves the separating DNA from other cellular components. Therefore involves separating DNA from other cellular components. Such as other cellular components, such as, of course, your proteins, RNA, and lipids. All right, so therefore you can say that DNA is a routine procedure to collect DNA for subsequent molecular or forensic analysis. Okay, so you can say DNA therefore, all right, is a isolation. Okay, so you say DNA isolation is therefore a routine procedure, is a routine procedure to collect the DNA. Why do we want to collect the DNA? We first need to isolate it for subsequent what? Molecular, basically, and what? Forensic. Molecular and forensic what? Analysis. Okay, so in our next going forward on this lecture, we want to look at how can this DNA be isolated. Okay, how can the DNA be isolated? How is it done? All right, so we want to continue this lecture by looking at the steps involved in the DNA isolation. All right, let me take this. So right here, let me put that as a subtopic. Steps involved. All right, steps involved in DNA extraction. Steps involved in DNA extraction, stroke isolation. Okay, so this could stand here as a sub-heading, and in looking at the steps, we say that DNA can be isolated the DNA can be isolated. The DNA can be isolated by placing the cells in a tube containing a special solution, and that solution is called a cocktail. All right, and it's mechanically called a cocktail, and mechanically or chemically breaking them up, and mechanically or chemically. These are two basic ways you can break up the DNA. Okay, mechanically and chemically breaking them up. Great. Therefore, the steps involved in the DNA isolation is as follows. Okay, so we say therefore the steps involved in the DNA isolation is as follows, or are as follows. So the first step we'll be looking at, let's say number one, is breaking the cells open. That's the very first step in DNA isolation, and this is commonly referred to as cell disruption, like we earlier said. Commonly referred to as cell disruption or cell lysis. And the purpose is to expose the DNA, within the cell. So that's the first step. Okay, exposing the DNA within the cell by what? Breaking it up. Okay, and we said this can be done through mechanical or chemical process. Okay, so how is this breaking of the cell achieved? This breaking of the cell can be achieved in different ways. Okay, so we say this is commonly achieved. Alright, this is commonly achieved. By grinding or sonicating. What? The sample. By grinding or sonicating the sample. All right, using different procedures. So it can be done using different procedures. Okay, grinding and sonicating the samples using different procedures. So to grind or sonicate the sample, you could use your mortar and pestle, you could use your blender, okay, to break and expose the cell. The step number two in isolating DNA from the cell is removing membrane lipids. Step number two, after breaking the cell, you remove membrane lipids. Remove membrane lipids by adding a detergent. By adding a detergent such as sodium toluene sulfate, your SDS. Okay, other forms of detergents you see you can add is your certin methyl ammonium bromide. Certin methyl ammonium bromide. Okay, and that is certin methyl ammonium bromide, CTAB, or your mixed alkyl trimethyl ammonium bromide. Okay, and that is your MTAB, mixed alkyl, MTAB. Okay, if you remove the cells and you add this detergent, what happens next is that it causes the cell to release its content into the cocktail containing enzymes, chemicals, and salts. So the addition of this detergent does the following. This causes the cell to release its contents into the cocktail containing enzymes, chemicals, and salts. Okay, so that's step number 2. By removing the lipids, you add a detergent. Example of detergents is the sodium toluene sulfate, certin methyl ammonium bromide, your CTAB, and your mixed alkyl trimethyl ammonium bromide, which is your NTAB. The addition of these detergents causes the cell to release its content into the cocktail containing enzymes, chemicals, and what? Salts. The step number 3, after doing step number 2, the next thing is to remove the proteins by adding a protease. Remove the proteins, okay, by adding what? A protease. A protease is an enzyme that can release or break down proteins. Okay, so this is, in bracket, this is an optional, and this step is optional, but almost always done. All right, this step is what? Optional. Though it's optional, it's always done. Okay, but almost always done. All right, then right there you have the 4th step of what? DNA isolation. The steps to DNA isolation. The next step we want to do is to precipitate the DNA. With an alcohol. Precipitate the DNA. Okay, we want to isolate the DNA and bring it out. So we precipitate the DNA with an alcohol, usually ice cold. Ethanol, or isopropanol, All right. So since the DNA is insoluble in these alcohols, it will aggregate together, giving a pellet upon centrifugation. Okay, and these steps also remove alcohol soluble salts. All right, so we'll take that down. The reason for using the to precipitate the DNA, precipitating the DNA with alcohol, the reason is because since the DNA is insoluble in this alcohol, which is your ice cold ethanol or isopropanol. Okay, in these alcohols, it will aggregate together, giving a pellet upon centrifugation, centrifugation. This step also removes alcohol related salts, okay, or alcohol soluble salt, removes alcohol soluble salts. Great. This is basically the steps in what isolating the DNA. Okay, so in the next slide, we want to begin to look at refining of these techniques, includes the adding of what chelating agents such as what your EDTA, which is your ethyl any diamine tetraacetic acid, in the extraction of buffer to chelate divalent cation such as magnesium and what calcium that are necessary cofactor for what the nucleases. This stops DNA's enzymes from degrading the DNA. DNA extracts often contain a large amount of what RNA, proteins, and what polysaccharides, tannins, and pigments, which may interfere with the extracted DNA and make it difficult to separate. So most proteins, by adding a protein degrading enzyme like proteinase K through denaturation at 65 degrees centigrade and precipitating using chloroform and isoamide alcohol, RNAs are normally removed using RNA degrading enzymes called RNase A. Okay, and polysaccharides like contaminants are, however, more difficult to what remove. So you have your common salt, your sodium chloride, together with your CTAB, is known to remove polysaccharides. So these are other components of the DNA that these steps refining the techniques just help us to remove. All right. So in the next slide, we want to see what happens next after the DNA is what extracted. Okay, so the DNA that is extracted will exist in long strands that form a mucus-like globe within the solution. Okay, all right, so the next steps after isolating, that's breaking down the DNA, okay, apart from what we saw in refining it, we want to quickly just take that along with these lectures. We do that in blue, sky blue. All right. So the DNA extracted, will exist as long strands that form a mucus, okay, that form a mucus-like globe within the solution. As it's within the solution like that, it contains a lot of what? Contaminants. So it comes with other compounds. So as the DNA is released, as the DNA is released, all right, as the DNA is released, along with other, as the DNA is released, along with other compounds like your lipids, along with other compounds like your lipids, your proteins, okay, lipids, proteins, you have your carbohydrates like we earlier saw. So all of these will be broken and they are all in solution. Okay, so it's released along with lipids, proteins, and all of this is broken and they are all in what? Solution. They are all in solutions. The carbohydrates, lipids, then you have things like or your phenols. All right, so all of this like this will be broken and they will be released in solution. Okay, so when they are released in solution, it needs to be separated from others. So all these are disturbing the DNA, therefore it needs to what? It needs to be what? Separated. Oh, let me do this in another line. Okay, so lipids, therefore the DNA needs to be separated. It needs to be separated from the others. What is others now? The lipids, proteins, carbohydrates, etc. Okay, so to do this, the DNA is harvested. So just know that it needs to be separated. In our next set of lectures, I'll begin by telling you how the DNA is eventually separated from all of these components. Thank you for joining us today at Biochemistry Simplified. My name still remains Dr. Elik, and I'll see you in our next set of lectures. Bye for now.